We shall proceed with the next session. Dr. Baldev Singh Oration. I call upon to the dais the chairpersons for this session, Dr. Satish Khadilkar. Dr. Nirmal Surya. Dr. Chandrasekhar Meshram. I request the chairpersons to start the session.
Welcome everyone for the Baldev Singh Oration. The speaker for this oration is Dr. Hannah Monyer from Germany, and her topic is how do inhibitory neurons control neuronal networks locally and at long distance, implications in health and disease. And to introduce the speaker, we have Dr. Suboor Bakth. Can we have Dr. Suboor, please?
Chairperson, Dr. Devika Nag, Dr. Satish, Khadilkar, and Dr. Surya. It is indeed an honor and a pleasure to introduce Professor Monyer, whom I know for several years. And I thank the organizers of IronCon, especially Dr. Chaudhary, for inviting us over and giving us the opportunity. To start with, Hannah Monyer was born in Romania and went to Germany at the age of 17. She finished high school in Heidelberg and studied medicine at the University of Heidelberg. After 5 years of training in child psychiatry and pediatric neurology in Mannheim and Lübeck, she went to Stanford in USA as a postdoctoral fellow with Dennis Choi. Subsequently, she did a second postdoc with Peter Seeberg at the University of Heidelberg, where she was a junior group leader. Since 1999, she is the head of Department of Clinical Neurobiology at the Medical Faculty of Heidelberg. And since 2009, she's also a professor at the German Cancer Research Center, DKFZ, of the Helmholtz Association. In her research on synaptic plasticity, learning and molecular biology, electrophysiology, optogenetics, and behavioral approaches, she is known for her achievements. She has obtained numerous prizes, including the most prestigious German award for scientists, the Leibniz Prize, which is the German equivalent of the Nobel Prize, and the Guy Lussac Humboldt Prize, the Sung Ming Tusu Prize of the Ministry of Science and Technology, Taiwan, the prize of the Berlin Brandenburger Academy of Sciences, and the Lautenschläger Research Prize. She's a member of the Heidelberg Academy of Sciences, of EMBO, the German National Academy of Sciences Leopoldina, and of the Academia Europea. She obtained 2 times the most prestigious European grant, the ERC Advanced Grant, that is funded with 2.5 million euros. And I must say, hers was one of the first labs in the world to introduce projection neurons. Indeed, she is a highly cited scientist and counted among top in the world in a field of excellence. No doubt, she appears frequently in media discourses such as newspapers, radio, and TV channels. Professor Monyer nurtures internationalization that is visible in form of diversity in her lab. In addition to her scientific excellence, Professor Monyer feels responsible for the society and actively takes part in various social initiatives such as supporting school and college students, introducing them to neuroscience, giving them the opportunity to visit her lab, nurturing their talent, and disseminating information to society at large. Hannah Monyer speaks 6 languages. She's an excellent pianist and a cello player and can be seen performing professionally. She's an avid traveler and very sportive, and some of her expeditions were to the Alps in 2022, to Mount Kilimanjaro in 2023, and to Patagonia in 2024. She is indeed a very kind and friendly person and takes personal interest in developing her team members and those associated with her. Last but not the least, she is a friend of India. So ladies and gentlemen, please join me in welcoming Professor Monyer on stage.
Thank you very much, Dr. Bakth. Thank you very, very much, Dr. Chaudhary, for inviting me to speak here at your, most important and yearly annual neurology meeting. As Dr. Bakth said, it's a privilege for me to be here in India for several reasons, not only to tell you something about our research, but also that I learn from you, learn more about your culture, learn about the diversity, but in particular to also learn what we have in common. And what we have in common suddenly is this thirst for more knowledge. You as a clinician and me, of course, as a basic scientist. As Dr. Bakth said, my background has been clinical, and it is due to 2 great teachers that in the end, I did my postdoc in Stanford, that I went into basic research. I do think it is very, very important whom we meet in life, in whose lab, in whose hospital we work, and where we are being trained, because these leading figures, they have a great influence on us. Now, I will delve directly into what we do in the lab, why we do it, what we know about inhibition in the brain, and why I and my colleagues think that this knowledge does have translational potential. I now just need to figure out where the pointer is. I see it here so that I can point. Yes, exactly. So about inhibition in the brain, first of all, I want to say that we work in the lab on learning and memory and how inhibitory neurons, what they do and why they are important in the hippocampus, the structure that is important for learning and memory. However, the principles that I will show you in the hippocampus, they hold true for many other brain areas as well. And this slide is just to remind me to tell you that how our memory functions. And when I say memory, I mean episodic memory. If you remember tomorrow that you have been here, if you remember the talks, it is episodic memory. It is what happens where and when. And for that, the hippocampus is of utmost importance. We come back to that. We read in Cicero's in philosophical works of the great orators of antiquity that people tried to enhance their memory by particular techniques, and I will show you one of them. But they understood already that memory is that it's very helpful for memory if you think and if you put what you try to remember in a particular place. What do I mean by that? So we read in these antique texts that if you try to remember something by heart, it is good to fragment that and put it on locations that are well known to you. So I will give you some examples. So this complex text is put on the digits of your hands. And you try to remember not only the text, but also to remember where you put it, to associate something that you want to remember with a particular location. And this association helps you later when you try to remember it to bring this back. Music can be remembered that way. This is a prayer. This is an incunable that I found in Karlsruhe from 1500. So this is a very long prayer that you say, and you put the different parts of the prayer. Now I'm lost the laser point. Again, on the digits of your hands. You can put it in a house, you can put it in a library, but it has to be known as a well-known space, and then you go back and retrieve it after having associated with that. These are examples from different literature over the centuries, but one of the best known is for the Europeans, Botticelli's representation of Dante's Inferno. So here the Inferno is represented as sinners. The greater their sin is, the deeper in the Inferno they are, and down here is the devil. So Dante traveled this Inferno in the literature, and he remembers the different sinners by associating them with the location where in the Inferno they had been. Now we go to what we do in the lab to try to understand how memory works and how inhibition actually works. So I was trained as a clinician, but in Peter Dieberg's lab, I turned to molecules, I turned to single channels. We try to figure out why particular cells have different channels. We see how these cells are embedded in a small network. We used also network modeling. This was a collaboration with Roger Traub for many years to actually then knock out these channels to try in behaving animals to see the contribution for learning and memory. So we go from the molecular to the cellular to the network level, all the way to the systems level at the behavioral level. Now let me try to explain to you the basic principle. And this is what my whole talk is about. And if you're in a rush to go for lunch, after this talk, you actually could leave. So this happens in the visual cortex. The principle is the following. The same cell can participate in different ensembles. So if we see in the brain, in the visual cortex, in the V1 region, we have feature detectors that detect round. We have feature detectors that detect color, for instance, red or yellow. Now, if you see a painting, and on this painting there is a lemon, the coincident spiking of neurons detecting round and yellow will be part of the representation lemon. Now, the same cell detecting round can fire again a little later, but now in the combination with a cell detecting red, it will be part of the representation of apple. And that happens all over in the brain. The same principled cell, in this case round, the feature detectors for round, will be part of different ensembles. And what I will tell you later is that it is the interneurons, the inhibitory neurons, that synchronize this activity at the millisecond range. And now let me use a metaphor. I use the metaphor of an orchestra. In the orchestra, you have different instruments. You have the violin, you have the cello, you have the gamba, you have the flute. So we work in our lab on the inhibitory neurons, whom I consider the directors of the orchestra. They do not code for so much, but they make it possible that these neurons that are the specialized feature detectors, that they fire with a millisecond precision And in this case, this neuron is part of one representation, and in the other case, it is part of another representation. And this principle holds true for all other brain areas. If you can move your arm, particular muscles need to be excited, others need to be inhibited. This needs to happen at the same time. And for this to happen, we need the interneurons that synchronize these networks. Now, here is again the principle in a cartoon. We have pyramidal cells. They are the excitatory cells. They are the music players. But then there are the interneurons, and the interneurons are the directors to make this synchronous activity in a network. Here we see a picture from Cajal, one of the fathers of neuroanatomy, as we all know. And you see pyramidal cells in the neocortex, and independently in which layer they are fairly uniform in their shape. Now, these are different types of interneurons, and they act in different networks at different rhythms. And the different rhythms you read as neurologists as the electroencephalogram. So we need these cells for this synchronous network activity that are represented in the EEG. Now, may I ask you what you see on this picture? Probably nothing, because in this picture, but I will ask you, what do you see in the next picture? You may see a dog. You may see, here is the head, here are the legs, here is the body. So already in 1920, 1930, the German psychologists, the Gestalt psychologists, they realized that it is our brain that binds together. We need to know something about the world. This is a top-down processing, and the brain binds this into a uniform representation. And here is the principle. If you see an image like this, our brain can read it either as two faces, and particular neurons are active in this type of presentation, or it can read it as a vase. And other neurons, if we record from that, and all this is known from monkey work, so the same picture can be seen either as this or as this. And it is the interneurons that bind these features together in a coherent representation. So this is the principle. The interneurons are the directors. The interneurons are the directors of this orchestra, and this is what we are interested in the brain. So this happens in the visual cortex. This happens in other areas. And let me quickly now turn to the hippocampus, the area that we are interested in. So for you as neurologists, you may know, this is known since the 50s of the last century, that the hippocampus is important for encoding something new. We know from Brenda Milner's work that the famous patient, HM, because of an intractable epilepsy, his hippocampi were removed at the age of in his early 20s. And this patient could not learn anything new. He had access to his old memories in the childhood, but he could not learn something new. What we know today is that we need the hippocampus for learning something new, but what we learn is not necessarily kept in the hippocampus. It is not stored in the hippocampus, but most likely in periods of silence, in periods of rest, and in particularly during sleep, it is transferred from the hippocampus into the neocortex. So that has been reproduced after the patient HM in many, many animal models. An animal can learn something, you learn that with the hippocampus regarding episodic memory, but then it is after a day, after two, after several days, it is transferred for storage in the cortex. You can remove from these animals the hippocampus, and the animal can still remember what it had learned, but it cannot learn something new. So in the patient HM, when the doctor came and talked to the patient, his intelligence was normal, but when the doctor walked out of the room, after 2 minutes, and he entered again, HM could not remember ever having seen this patient again. So this is what the hippocampus is doing, and that is where we record. Now, these are two images from Heidelberg, and the same principle holds true what I have told you already. This is the castle in our city. This is the river, the river with the old bridge. And it is the interneurons who dictate that, for instance, this black neuron is part of this ensemble in this case and is part of the castle. And in the next, you go across the river or you remember this, the same neuron can be part with the other gray cells of another representation. And it is the interneurons who actually help generate either this representation or that representation. Now, and this is done again if you are a neurologist in the hippocampus when one walks or when one actually has a particular exploratory activity. We read this as theta rhythmic activity. Also, when I explore the environment, we would have theta rhythms in the hippocampus and gamma activity on top of it. Now, in the animal, we have exactly the same rhythms. So when the animal explores, you have synchronous theta. When the animal is still or during sleep, you have a very different activity. We can, from the activity of the animal, deduce actually the behavior because it is typical for that particular behavior. Now, in the lab, we record this by implanting tetrodes where you don't see only the rhythmic activity, the EEG, which gives you information about the whole network, but individual spikes. So we see the musicians at work. We see which neurons we can, with particular programs, decode which neuron has been active where and when. And again, just to compare the human brain and then what we do. So this would be time and space showing that when recording with implanted electrodes, we have a resolution which is with a very fine resolution in the micrometer range and with very, very fast with temporal resolution in the millisecond range. And you as clinicians know that, of course, we get from patients information, by looking at the brain, by MRI, by different means that are non-invasive. You have a good spatial resolution, a good temporal resolution, but it is not comparable with what you do by inserting tetrodes in the brain. And you as clinicians know, in clinics, this happens maybe in epileptic patients, for instance, before surgery, but also for tumor patients to look, the healthy from the affected brain region. Now, I want to show you the neurophysiological basis of memory, as has been shown by a number of different labs. So we recorded these tetrodes, we record the rhythms, the animal explores its environment, and it was O'Keefe who, together with the Mosers, got the Nobel Prize for the discovery of place cells. Place cells are cells in the hippocampus that fire when the animal runs around in a very particular location. We represent this as a heat map. So cell number 1, so red is high activity, blue is very low or no activity. Another cell would fire here, we find a cell fire there, or here. When you put the animal after 20 minutes in the same environment, these cells are stable and they fire in the same location. So in the hippocampus, and I will show you the other brain regions, we have a GPS system, if you want so, and if I walk from A to B, the same cells will be active again and again in the same spot. And I represent space by the activity of these neurons. So this is a particular coding in the hippocampus. Now, the region that feeds into hippocampus is the medial entorhinal cortex. And there the Mosers found other cells that are spatially tuned. Here again would be a place cell in the hippocampus. So by the way, here in black, you see the trajectory of the animal, and every red dot is a spike, is an action potential. So this cell fires in this environment, in this location. Now the Mosers found in the medial entorhinal cortex in cells that fire in multiple locations. So again, these are not different place cells, but one and the same cell that fires in multiple locations. And you can see that if you unite basically the apex of these cells, this gives you a grid pattern. And it was in our lab we showed that the grid pattern or the grid cells are another part of the GPS system. What they measure is the distance that an animal traveled and also path integration, and I come back to that. There are also border cells, so these are cells which fire only at the border at the wall of an environment. Thereby, one hangs up, one fixes this cognitive map. So our hippocampal formation, so the hippocampus proper and the medial entorhinal cortex, they build a cognitive map of the environment, okay? And what we also have in the adjacent region are object cells, so this encodes for space, and then we have cells that code where particular objects are located, and yet other cells where the object has been previously been. So in the hippocampal formation, you have highly specialized cells that code for what happens, where, and when. What is the object cells, and when and where is the spatially tuned cells. Here an example from our department, from Kevin's recordings. You will see here the animal running. This is just a cartoon. In 20 minutes, the animal, so every dot is an action potential of the same neuron, and after 20 minutes, the animal has covered the whole environment, and you have, this would be a place cell in the hippocampus, and this would be the grid cell, measuring the distance the animal has run, if it runs on a straight, or just mapping the space during this exploratory activity. Now, as I showed you before, or we know actually for sure that these interneurons are the ones, I showed you the place cells and the grid cells, they are excitatory neurons. But what we are interested in is how these individual interneurons, the directors of the orchestra, how they conduct this orchestra. And you can actually tell from now, actually, if we change their activity, the whole orchestra will be out of tune. So what I will show you now is, in fact, if we do genetic alteration just of the interneurons, our memory will be altered. Here are two examples, anatomical examples of filled neurons in the hippocampus. And what you can appreciate is these interneurons, they stay locally. They affect just a local network. Such an interneuron makes about 20,000 synapses in the brain. Here another interneuron, again in red, the cell body or the dendrites. Here goes the axon. It ramifies and it stays in the region of the dentate gyrus and controls the networks of the dentate gyrus. Now, I do not have time to show you here, but this is work, the whole field of interneurons goes back to the 60s of the last, so we are talking only about 50, 60 years of research. And this is particularly work of Tamas Freund and of Peter Shomogyi and many other labs, including ours, characterizing different types of interneurons and how what they do in the brain, how they are connected to which cell types and which rhythms in the brain they control. So as I said, in the hippocampus, you have mainly theta and gamma. In the visual cortex, as clinicians, you know, you have alpha rhythms. There are certainly different interneurons directing these networks, but the principle is always the same. It is different pyramidal cells that participate in different ensembles that are recruited by the interneurons. Now, for the aficionados amongst you, and this is, however, very important for the clinicians, is that the different types of interneurons are all generated like all other cells in the brain during embryonic development. And where they come from, here this is now from a mouse brain, but the same holds true from the human brain. For instance, the parvalbumin, fast-spiking cells, they come from the medial ganglionic eminence, the calretinin, another type of cell, they come from the lateral eminence. So it is dictated by the place of birth. There is a genetic determination where these neurons will go after migrating into the cortex. And you may not know as an adult neurologist, but you know, as a pediatrician and neurologist, there are a number of neurological diseases that result from defaulted migration during embryonic development, but also diseases in psychiatry such as autism and schizophrenia have been associated with faulty migration. and then the disease develops at a later stage. So there is lots of research. My lab does not do so much in development. We have a couple of papers on this as well. We are also interested in GABA signaling. Why is that important? As you may know or may not know, during embryonic development, but also shortly after birth, excitation in the brain is provided by the inhibitory neurons. So you should not give a pregnant woman benzodiazepines, because the influence on the baby will be excitatory, not inhibitory. Okay, so knowledge about such or basic knowledge is very, very important for clinical use. And this is also something that we've understood only over the last 20 years or so. So because GABA is excitatory very early in development, not inhibitory. It becomes inhibitory. If you're interested, we can talk during the break why that is the case. Now, as a young postdoc with Peter Seeburg, we cloned many glutamate receptors, and you probably know that the glutamate receptors are the excitatory receptors in the brain. And what I found out as a postdoc was that interneurons, very differently from pyramidal cells, have different receptor subtypes. Again, these are genetic programs. And why do I know this? Because I performed single cell PCR. So in acute slices, you can identify interneurons because their firing pattern is differently from the excitatory cells. You can take out the content of the cell. You can do gene RNA expression analysis. And we found that these glutamate receptors in the interneurons are much faster than in pyramidal cells. So they open faster, they close faster. And we then did, in my own lab when I became a junior professor, we did mice, or we generated mice where we knocked out these particular subunits. So you can still recruit the interneurons, but not as efficiently. And what is shown, I show you just a very simple test, how we analyze this. And this is what you think of when you think of short-term memory. If I tell you to remember a number, 25183, you remember it for a very short time. This is done, in this case, with the hippocampus. But if this is not repeated, the animal will not remember it. So how does this test work? I hope that I'm supposed to be able to activate it. It worked before. Where is the young man who was supposed to help me? He promised he would be here. Okay, he went for lunch. Okay, this movie. So what the mouse should do, the mouse runs in this arm, and this arm is closed. And the mice, if they feel secure, can you activate it from there that it runs? No, my laser pointer should stay. I explained the test to you. It does, don't worry. I just told him he should be here. Just bring it back. I can explain it. We are losing time. Okay, so the mouse runs in this maze, and it's called a T-maze, and here it gets some sweetened milk. The mouse does this for the first time, so it's not a test. It's just used to the room and to this, but it's not something that it has learned. It just feels comfortable. And you will see when we put the mouse back, it drinks the sweetened milk here at the end of the arm. You put the mouse back, and if it has a good working memory, and now you open this other arm, the mouse will run because it is curious. It will run into the other arm, given that it remembers where it has been before. The mice, like humans, they have this tendency to go to what is novel if they feel comfortable, right? So, and this is measured here in black. These are the knockout mice where we generated by knocking out one of these two important subunits. And what you can see is that the mice are at chance level. So bottom line, we do not change the activity of the pyramidal cells. We just change the way by which interneurons are recruited. And the next slide, which I now can't activate from here. Can we go back into the, thank you very much. Now, okay. got stuck. Okay. The next slide. Bit from there, please. Okay, I will, no, it's too much. I will skip it. Don't worry, don't worry. I will skip also this because otherwise we're running out of time. Okay, I'm coming back to the other. So what, I had a few more examples to show you that if you fail to recruit the interneurons properly, you have a working memory deficit. And that happens in different brain regions. You have working memory in the visual cortex, you have working memory. So if you keep something in your visual memory, and we are specialized on the hippocampus where we show this. Now let me go to one of the, I would say, one of the most important discoveries which we made in the lab, and that was Sarah Meltzer and Antonio Caputi. Now, you have heard when I speak about GABA cells, I say interneurons. It was believed that in the neocortex and in the hippocampus, all GABA cells are interneurons. That means they do not project out of that region. They control the local rhythm. But as neurologists, you must know that for many things, we see and hear something together. The right brain hemisphere and the left hemisphere have to be coordinated. Now, the conduction velocity of the axons is way too short to account for synchronous activity in brain areas that are at such long distance. So just by chance, we actually found that in this population of GABA cells, there is a small percentage of neurons that are actually projection neurons, and they coordinate the activity at long distance. So let me try to explain this. And now you don't see, wait, we go to the pointer again. Okay, so what I had explained before is that we have interneurons, the red cell, controlling one rhythm. This, let's say, is an hippocampus, and you measure this as synchronous activity in the theta range. Now, if you have a brain area that is further located, like the medial entorhinal cortex or the septum or the cortex, you would have, depending on the task, different activity, different oscillations in your EEG. Now, but these areas, they do not communicate or they cannot be synchronized in the millisecond range. So what we found in the lab was that there is a particular type of GABA cell that projects at long distance, and at long distance, it inhibits selectively the interneurons of the other ensemble. Thereby, it synchronizes areas that are long distance, and it contributes to information flow from A to B. And these neurons we found in many brain areas. We found them between different cortical areas. We found them between the hippocampus and the medial entorhinal cortex. I do not have time to go into this. And this would be the data. So this was Antonio injecting a particular virus, and this virus expresses a fluorescent protein in the interneurons of the medial entorhinal cortex. This is the green area. It is the main input to the hippocampus, where all the sensory information from the entire brain come and are actually transmitted to the hippocampus. And you can see now in the hippocampus, these long axons, which nobody had seen before. Actually, it's not true. There is some anatomical work by Eberhard Buhl, but nobody believed that because, you know, the normal dyes, they can actually go also at far distance. So what does this bring us? What advantage? So we use a technique which now is generally termed optogenetics. You can bring, by viruses or transgenic methods or other methods, you can bring particular channels into neurons which can be activated by light of a particular wavelength. So in other regions, in other words, you inject the virus, and then with this blue light, you activate this particular neuron. So what is shown here as a schema, you inject in one brain region, and then with a patch pipette, we measured, this is now in the medial entorhinal cortex, and Sarah measured in the hippocampus. With blue light, we activate that cell, and we find the responses, and these responses are purely GABA responses. Now, This is the principle of long-range inhibition. Inhibitory neuron inhibit other neurons at long distance and disinhibit that brain region. Now, many projects in the lab actually deal with the pacemaker of the whole hippocampal formation, and that is the medial septum. The medial septum is a subcortical region, and as we have pacemakers in the heart, in the brain, for the entire hippocampal formation, the medial septum is actually the pacemaker of this major rhythm in the hippocampus, and it does different things. It generates theta, it generates burst firing of the neurons. This is too far to go into this, but all this can be studied with optogenetics. In the slice preparation, you inject the virus here, you make a slice from this brain region, you cut, and the slice is alive for 5 hours or so, and you can see how these cells communicate. You can also use it in vivo in freely moving mice by implanting a fiber and optogenetically stimulate the cells of your interest. Now, let me finish again why also from a clinical point of view, the hippocampal formation is very important. And for that, I need to introduce you to the last topics. I have just a few more slides. I have mentioned these cells, the grid cells in the medial entorhinal cortex. Now, what we showed, but this had been predicted by modeling already, but it was in our lab that Mariana Gil showed this for the first time, and that these cells can do path integration. So what is path integration? We humans can do it, the animals do it, vertebrates. So when an animal runs, even in the dark, runs zigzag in an environment, the animal comes straight back more or less to where it had been. This you can do yourself, If you cannot do it, you should see a neurologist. So this is path integration. And this is something that is not a learned behavior. That is something that is inherent. It gets better. We build a representation of the world in our brain. We have learned that, of course, but it is dependent on the movement of our body. And it's known from lots of work, particularly from Jeffrey's lab, if you put an animal on a little wagon and you pull this, so if the animal does not move itself, all the spatially tuned cells are not as precise as if the animal moves. So you have to move yourself in order to build this cognitive map. And the animal can return from here. This is now shown that we in humans, we do the same, So if this is an outward path, we are actually able to come back fairly straight to where we have been. Now, in normal life, we do not use it when we have a visual cue. When we have a visual cue, we usually depend on that, but we use it in particular circumstances. Now, why is this important? This is important, as you know, as clinicians, the hippocampal formation is amongst the first to be affected in Alzheimer's disease. And amongst the first symptoms that patients have is they have memory impairment, but they also get spatially disoriented in the environment. And as I have told you now over the last 50 minutes or so, the hippocampus does both. The hippocampus is important for memory formation, and the hippocampus is important for spatial tuning. All our memories, all the sensory input is put together with the information where we are as an episodic memory. And here just one example to show you. This would be, for instance, a normal grid cell in a mouse, in a wild-type mouse, and this would be a mouse. There are many genetic mouse models that are affected this way. Now, again, as a repetition, how does this happen? I had shown you, you have these individual neurons. They represent different things. We come back to the hippocampus. And you can measure either in this or in this environment your particular rhythms. And if you have a long-range neuron, then you would have a connection between these brain areas, which helps the fast information flow. Here again, examples from our lab now that is medial septum neuron, which are GABAergic projection neurons and are located in the septum. This would be their axons to the hippocampus, to the medial entorhinal cortex. This would be from a wild-type mouse, and this would be from 2 models of Alzheimer mice. Now, as you probably can expect, in Alzheimer pathology, lots happens in the brain. But what we found in our lab is that these pacemaker neurons are amongst the first to be affected. And why do we think that they are first to be affected? Because these cells fire at a high rate. They need lots of ATP. And indeed, what Elke Fuchs found in the lab is that the mitochondria at very early stages, when the rest of the brain is not affected yet, that the mitochondria in the axons of these cells are already fragmented. So I come to an end of my lecture. Let me briefly summarize. I told you that GABAergic inhibitory neurons are the controllers of rhythmic synchronous network activity. I have shown you that these cells, that most of them control local networks. If I see something, it is the visual cortex that is affected. But if the environment is very complex, if different networks are active, then we need synchronous activity between these different networks. And we have good reason to believe that these long range, which I call super directors, because they direct the local directors, okay, it's a very parsimonious and a very precise way of basically synchronizing networks at long distance, that they are very important and for many, many cognitive processes in the brain. And finally, what I've shown you, because these cells are actually very, very active, that I have not shown you, but that we know, we think that they are very vulnerable at very early stages in Alzheimer's diseases. And by the way, it is known that in many other neurodegenerative diseases, such as Parkinson, such as Huntington, such as ALS, mitochondria and fragmented mitochondria are amongst the first symptoms or the first signs that you can see at a cellular level. With that, I come to an end. Our lab is actually a fairly big lab, but what I showed you today as a recapitulation was done by the people that I have mentioned along the way. I had great collaborations over the years for Miles Whittington, Roger Traub, and Peter Jonas. And these are my 2 teachers who actually have influenced me a lot. From Dennis Choi, I got into science. I learned the beauty of basic science. I developed the passion. And when I went into Peter's lab, there I developed an addiction for science that did not let me go anymore. So I'm very, very grateful. All this work could not be done by me alone, but by many talented people in the lab. And this has been generous funding over the years from many agencies, in particular for the German, from the DFG, and of course the ERC, but also other institutes. Thank you very much for your attention.
Thank you, Dr. Hannah. It was a wonderful oration, and it was a great tribute to the great scientist, Professor Baldev Singh. I had the privilege to interact with him during my days at PGI Chandigarh. He used to visit the institute when I was doing MD and DM neurology and take bedside clinics. And what we realized that he was ahead of time in thinking as far as the neurobiology is concerned, and he has a aptitude for research, and he is doing a great research. So it's a great tribute to the wonderful researcher and neurobiologist of the India, Professor Baldev Singh. Thank you so much.
Thank you very much for the kind words.
I request chairpersons to felicitate orator Dr. Hannah.